Okay, well, I'll go ahead and get started then. So, yeah, for anyone who may not know me, I am Seraphina. I'm a postdoc in Dr. Emily Reese's lab, and I'm in my third year of my postdoc now. So I'm really excited to share some of the work I've been doing over the past couple of years. And I'm going to focus today on sharing some of the work I've been doing looking at the blood-brain barrier using really cool neuroimaging techniques and different markers of Alzheimer's disease risk. So I really like this picture here that I've shared on my title slide that shows how dense and complex the brain vasculature is. So much so that you can kind of even make out the macro anatomy of the brain just by looking at the vasculature, which is kind of a nice reminder of how kind of central and important of the role it plays in brain health and aging. So here is a brief agenda of what I'll be talking about today to follow along in the style that some of our other Lunch and Learn presenters have been using. I'm going to start by giving an overview of my personal educational and research history and how that's brought me here, and comment a bit on some of my future goals. And then I'm going to share two projects for my postdoc work. One that was recently published, actually, looking at blood-brain barrier permeability, heterogeneity, spatial heterogeneity across the brain, as well as differences by ApoE4 genotype and relationships with brain microstructure in cognitively normal older adults. Then I'm going to share some more recent work that is still kind of preliminary. I just presented it at the Alzheimer's Association International Conference, looking at associations between blood-brain barrier permeability and modifiable lifestyle factors. And then lastly, I will end by sharing some of the insights I've gathered along the way and some advice that I have for other trainees. So starting with a little bit about myself and my background, I've actually lived in the San Diego area for a very long time. I've been here since I was 9 years old. So at this point, most of my life has been spent here in San Diego. And I went to college at San Diego State University, where I double majored in biology and psychology. And I worked there in the Brain Development Imaging Labs, or BDIL, with Dr. Ralph Axel Mueller, and my advisor that I worked most closely with was Dr. Ruth Carper. And this lab studies autism spectrum disorders. And my work in this lab and my experience in this lab was really rewarding, and it was really foundational and has influenced my research interests over time. My honors thesis in this lab was looking at the impact of head motion during scanning on findings of group differences in diffusion MRI measures. And I'll talk a little bit more about diffusion MRI in a bit because we use that tool in the Reese lab as well. But head motion, the role of head motion, this isn't something that's specific to autism research. It has broader implications. And so this early methods-focused research has really stuck with me and shaped my interest to this day. Another really valuable experience that I got while at SDSU was some tutoring experience. So when I first started college, I was working as a tutor for the AVID program. AVID is a program for middle schoolers and high schoolers that come from underrepresented or disadvantaged backgrounds to help them prepare to go to college. And I was a member of this program in middle and high school, and then got to work as a tutor for them when I was in college. And so this was my first experience working in an official capacity in like an educational setting to help other people learn, and it was really rewarding. Then a couple years later, I started working as a tutor at the Fowler Athletic Center at SDSU. And so there I was helping college athletes with their classes on a wide variety of subjects. And so by the time I graduated college, I had two tutoring experiences that I really, really enjoyed. And these kind of planted the seeds in my mind that teaching was something that I might be interested in as well in my career. I also wanted to share that I was a part of the NIH IMSD program at SDSU. So IMSD is Initiative for Maximizing Student Development. It's a T32, a training program that's kind of the college version of AVID. So it's meant to help students from underrepresented or disadvantaged backgrounds prepare to go directly into graduate school after undergrad. And so both of these programs were so immensely important for me, and I credit a lot of my progress to the help that I got in those programs. The last thing that I'll mention is that in my last year at SDSU, while I was applying for graduate programs, I applied for the NSF GRFP, the Graduate Research Fellowship Program, and got an honorable mention. So in 2017, I came to UCSD in the Neurosciences graduate program. This is where I did my PhD. And I actually had a joint mentorship between Dr. Larry Frank and Dr. Mark Bondi, who many of you here know. So Larry Frank's background is more in physics, and his work focuses a lot on pulse sequence development and development of MRI analysis tools with a special focus on diffusion MRI. And then Mark Bondi is a clinical neuropsychologist with expertise in cognitive aging and Alzheimer's disease. So it was really cool to get this very interdisciplinary training, getting experience with both the hands-on nitty-gritty methods side of things as well as the cognitive and clinical side of things. I reapplied for the NSF GRFP in my second year as a graduate student and did receive that award. I recently learned that this year they are no longer allowing second-year students to apply. I'm very sorry to those who may have been affected by that decision and don't get the chance to apply for that award. And in grad school, I continued to get some teaching-related experience. I volunteered with the Neuroscience Outreach Program and some other community events and was a teaching assistant for an undergraduate neurobiology laboratory class here. So I was still very interested in both research and teaching at this point. So then in 2023, I started my postdoc working with Dr. Emily Reese. And I am sharing here some of the other grants and funding mechanisms that I've applied for, mostly because I want to emphasize a lot of the opportunities that are available here at UCSD, whether that's through the Alzheimer's Disease Research Center and also the Stein Institute. So shortly after I started my postdoc, I applied for the NIH NRSA F32 award, which I did not get. And then 3 UCSD ADRC grants, the T32 training grant, the REC award, and the development award. And I did receive the T32 and the REC awards, which I think have options both for graduate students and postdocs, or anyways. More recently, I applied for the Bright Focus Fellowship, which has awards for postdocs doing Alzheimer's disease research, and the Stein Institute for Research on Aging, Healthy Aging Research Program, which is another really excellent resource for those who are doing this kind of work. I also wanted to mention my REC award my mentor for that award is Dr. David Salmon. And that's been a really excellent experience as well, getting to do another side project on top of my work in the Reese Lab. And then lastly, I'll mention here, I know I'm taking a long time with the background, but when I started my postdoc, I also got this amazing opportunity to teach a class at San Diego State University. So I got to take over this course on neuroimaging, and totally designed my own curriculum. And I'm teaching that for the third time now. And that's been a really amazing experience. And at this point has really solidified how much I do enjoy and value teaching. And so now my career goals, what I'm trying to keep an eye out for as I am looking for next steps, is to find faculty positions that have a nice balance between research and teaching. Okay, so now I'll get into some of my postdoc work. So I want to start by introducing the blood-brain barrier. So the blood-brain barrier is not a single physical barrier separating the vasculature from the brain tissue. It's a complex set of anatomical and physiological characteristics that limit permeability and regulate the transport of ions, molecules, and cells between the vasculature and the brain parenchyma. I like this definition because it emphasizes that the blood-brain barrier is this collaborative community of anatomical and physiological characteristics, and it plays a really big role in making sure that the brain gets what it needs from the blood, nutrients and oxygen, and that potentially dangerous things are kept out, and also that metabolic waste products can get cleared out of the brain as well. And because this is such a complex set of characteristics that build this barrier, there are many ways that it can go wrong. And in fact, blood-brain barrier disruption is implicated in early Alzheimer's disease. So neuropathological studies show blood-derived proteins in the cortex of patients with cognitive impairment and Alzheimer's disease, suggesting that the blood-brain barrier has been damaged. And molecular marker studies have found CSF markers of BBB abnormalities in these patients as well. So both of these pieces of evidence suggest that something's going wrong with the blood-brain barrier early in Alzheimer's disease, but each approach has some limitations. So with neuropathological markers, we can get good anatomical localization of blood-brain barrier disruptions, but we can only get these measures at the end of life. And with molecular markers, we can measure them in vivo across the lifespan, but they don't give us the anatomical information. So there is a method that we can use to get the best of both worlds here. That is DCE MRI, which I will talk a little bit about now. So DCE MRI stands for Dynamic Contrast Enhanced MRI. So it's a dynamic scan, meaning that we're collecting data over time. So we're collecting repeated T1 anatomical scans before, during, and after injection of a gadolinium-based contrast agent. And then we can model the uptake of that contrast agent into the brain tissue over time. So this is a process called pharmacokinetic modeling. And in particular, we use the PATLAC model, which assumes one-way flow of contrast agent from the blood into the brain tissue. And this gives us a measure called K-trans, which is essentially a measure of blood-brain barrier permeability. So higher K-trans means that the blood-brain barrier is leakier. I will also note here that the K-trans measure, in fact, is a little bit more complicated than that. It's influenced not only by permeability, but also by blood flow and vessel surface area. So the reason I mention this now is because this idea may play into some interpretation of one of my findings that I'll share later. Okay, so now I'll show you what a K-trans map of the brain looks like. Scrolling from superior to inferior view in an axial view. I don't know how well you're able to see that on the screen, but the permeability values we're looking at here, they're very low. We're not looking at major leakages like what we might see with a stroke. We're looking at very subtle, small scale leakage here. And it's only been until relatively recently when we've had the imaging and the analysis tools that were sophisticated enough to let us actually measure this really subtle signal. So some DCE MRI work previously showed that the blood-brain barrier was more leaky or more permeable in people who have the ApoE4 allele, which is the strongest genetic risk factor for Alzheimer's disease, as well as in people with mild cognitive impairment and Alzheimer's disease. However, these findings differed based on whether the effects were localized to specific regions like medial temporal lobe structures, or if they were more widespread across the brain. So this is what motivated a lot of the recent work in Dr. Reese's lab. And before I get into my project, I'll quickly share some results from a project that was published in the lab last year, where we were trying to ask this question: where is blood-brain barrier leakage restricted to these medial temporal structures, or is it more kind of global? So what we found, first of all, was that ApoE4 carriers had leakier blood-brain barriers than non-carriers in the global gray matter, so averaged across the whole cortex, but not in specifically these medial temporal structures, so entorhinal cortex, hippocampus. We also saw relationships between blood-brain barrier permeability and measures of brain microstructure among higher risk groups. So I think that this figure might be difficult to see, but what we're showing here is that in the entorhinal cortex, higher blood-brain barrier permeability was associated with lower restricted isotropic diffusion, lower neurite density, and higher free water, all among amyloid beta positive individuals, and then also associated with higher free water among ApoE4 positive individuals. So these microstructural markers are indicators of early neurodegeneration. So we found these patterns and we saw that this seems to be a global effect. And so what I wanted to ask next was, okay, well, what if we look at the cortex more divided into its different areas? Are there interesting anatomical patterns that we might uncover? So then I wanted to ask, okay, well, maybe instead of using these global measures, global gray matter, global white matter, what if we break it down into individual regions? Might we see some interesting spatial patterns there? So that's what I looked at here in this paper that was just recently published. So going into a bit more of the methods, we collected 3 types of MRI scans for this study: dynamic contrast enhanced DCE MRI, which I've already described, T1 weighted MRI, which we use for co-registration and anatomical parcellation in FreeSurfer, and then restriction spectrum imaging, or RSI. RSI is a diffusion MRI technique, and diffusion MRI is what lets us look at brain microstructure. It does this by measuring water diffusion or water movement in the brain, and depending on the pattern, the magnitude, and the timing of that water diffusion, we can take the signal coming within a voxel and divide it up into the fraction of diffusion that's coming from restricted spaces like inside cells versus hindered spaces, which is the extracellular space, or free water like the CSF. And we can also divide the signal up into whether it's coming from anisotropic compartments or isotropic compartments. So isotropic is like a spherical shape. So this might be things like cell bodies. And then anisotropic is like basically not spherically shaped. There's some directional component to it. And so this is going to be more like neurites, axons, and dendrites. So we can get really valuable microstructural measures like neurite density, free water, and other measures. And I'll explain them a bit more as I introduce them in the findings. So in terms of image processing, we used a program called Rocketship to process the DCE MRI data. This is the tool that lets us calculate the K-trans, the blood-brain barrier permeability across the brain. And then with the T1 images, we used FreeSurfer to do cortical gray matter parcellation, so dividing up the cortex into all of these Desikan, Kiliani, Atlas regions, as well as subcortical segmentation. And then we also looked at the white matter in regions defined using the Atlas track atlas. So the whole brain, we looked at gray matter, white matter, and subcortical regions. And we averaged permeability values across the left and right hemispheres. And so we had a total of 34 cortical measurements, 8 subcortical measurements, and 13 white matter measurements. We also looked at ApoE genotype. So we classified people as being ApoE4 carriers or non-carriers, and also classified people as being amyloid beta positive or negative. And for the amyloid beta classification, we used 3 different approaches depending on what data were available. So in order of priority, given data availability, if they had PET imaging, we used that to classify amyloid positive or negative. If they didn't have PET imaging, we would next prefer CSF, cerebrospinal fluid. And if they didn't have CSF, which was 4 individuals, we inferred amyloid beta positivity from plasma p-tau 181. Okay, here are our participants. So we had a total of 48 participants. 44 were from the ADRC, and then 4 participants were from the San Diego community. We had 25 ApoE4 carriers and 23 non-carriers. And these ApoE groups did not differ in terms of age, years of education, total MMSE scores, the number of women versus men, or the number of amyloid beta positive individuals. Okay. So the first thing that we found was that blood-brain barrier permeability varies across the brain in cognitively normal older adults. So it's not this homogeneous measure. So we can see here on the left, this is a cortical map. So this is showing average permeability within each of those Desikan, Kiliani cortical regions. And we can see here that, there's different regions that show higher versus lower permeability. In particular, we tend to see higher permeability in these inferior temporal and frontal regions, as well as some sensory areas. Subcortical maps are shown in the middle panel, and then white matter permeability averages are shown on the right. We didn't have a good way to visualize concisely all of the white matter regions. What we can see here, so a couple things. A, blood-brain barrier permeability does vary across the brain, and B, tends to be higher in the cortex than in the subcortex in the white matter. Okay, so the next thing that we found is that ApoE4 carriers show higher blood-brain barrier permeability in several cortical regions. They show significantly higher permeability. Let's see if I can read these. The bolded names here: the cuneus, paracentral lobule, precentral gyrus, inferior parietal lobule, and caudal middle frontal gyrus. And in fact, after we further control for amyloid beta positivity, not only do these effect sizes get stronger, but some additional regions come out as showing significant differences between groups. So this ApoE4 pattern of higher permeability also varies by brain region and seems to be independent from amyloid beta status. So what people might be wondering now is, well, why do we see these specific regions showing these ApoE4 differences? Why do ApoE4 carriers have higher permeability in, the precuneus, precentral, whatever, gyri? So one current hypothesis that we have that is a bit speculation because we can't really determine this with certainty with the data that we have. But one idea is that it might relate to vessel density. So remember that I mentioned K-trans is influenced not only by permeability, but also by vessel density and by blood flow. And also if there's like a much higher vasculature in the cortex, then there's just by nature going to be also more permeability. So this figure here that I'm showing is from Seraphina et al. 2024, where they used a type of MRI called black blood MRI, which lets us look at vessel density, cerebral vessel density. And they were comparing younger versus older adults. The younger adults were under 35 years and the older adults were over 60 years. And so these are the regions where they found that the older adults had significantly lower vessel density compared to the younger adults. And there does seem to be this rough correspondence between these maps of this kind of like pattern along this midline gradient from the posterior to anterior regions, which is kind of similar to what we see with these ApoE4 effects. And there's been some rodent work showing that mice with human ApoE4 show reduced blood flow due to is related to impaired neurovascular reactivity and renders them more vulnerable to damage from reduced cerebral blood flow. So I had just finished talking about the ApoE4 differences in blood-brain barrier permeability and how one current explanation for the spatial pattern that we see could be related to vascular density. So now here are some findings where we were looking at associations between brain microstructure and blood-brain barrier permeability. And we saw this interesting pattern similar to the previous paper where blood-brain barrier permeability does correlate with brain microstructure, but specifically among 3 groups of people who are at higher risk for Alzheimer's disease. So ApoE4 carriers, amyloid beta positive individuals, and women. So what we found, looking on the left panel first, is that in the rostral middle frontal gyrus, higher blood-brain barrier permeability was associated with higher free water and lower hindered diffusion among ApoE4 carriers, but not non-carriers. So these patterns, higher free water and lower hindered diffusion, may be indicative of neuroinflammation. And that next, we saw that in the entorhinal cortex, higher blood-brain barrier permeability was associated with lower neurite density and lower restricted isotropic diffusion among amyloid beta positive individuals. So lower neurite density and lower restricted isotropic diffusion may be relating to loss or shrinkage of neurites and cell bodies, so early neurodegeneration. And then finally, in the superior frontal cortex, higher blood-brain barrier permeability was associated with higher free water among women, but not men. Again, possibly indicating neuroinflammation. So this relationship between blood-brain barrier permeability and microstructural patterns that could be indicative of early neurodegeneration in high-risk individuals suggests that this permeability may be relating to the heightened risk for AD. Okay. So that was the first project. So this next project that I'll go through kind of quickly was more recent. Again, this was just presented at AAIC, so these are more preliminary findings, and I very much welcome feedback, questions, suggestions on this. So here we wanted to look at modifiable lifestyle factors, and in particular, lifestyle factors involved in vascular health. So we looked at alcohol consumption, smoking, physical activity, and sedentary behavior. So this sample, we had 53 cognitively normal older adults, 26 women from the ADRC and San Diego community with an average age of 75.8 years. So for alcohol consumption, we classified people as being abstainers, meaning they have not had any alcohol within the past year, but we don't know much more history beyond that. Moderate drinkers, which is defined as people who have up to 7 drinks per week or 1 per day. And then a heavy drinker was classified as people who have over 7 drinks per week or on average more than 1 per day. So you can see our groups here, 7 abstainers, 39 moderate drinkers, 7 heavy drinkers. Our abstainers and heavy drinking groups are very small. So keep that in mind. For smoking, we looked at it in 2 different ways. First is a binary variable. Have they ever smoked or have they never smoked? And we had 22 ever smokers and 31 never smokers. And then we also looked at a continuous variable of lifetime smoking exposure or smoking intensity, which was calculated as the product of the average number of cigarettes smoked per day times the total number of years smoked. So we were trying to look at there, like, does it matter how much someone smoked rather than just simply whether or not they ever smoked? Then for physical activity, we looked at whether people reported that they engaged in regular strenuous exercise and exercised at least 3 times per week. So if they said yes to both of these things, we categorized them as regular strenuous exercisers. And if they said no to at least one, then they went into the no group. This classification gave us really equal group sizes in each. And then we also looked at the average number of hours spent walking per day. And for sedentary behavior, the average number of hours spent sitting per day. So all of this information was self-reported via questionnaires. So for alcohol, here's what we found. Heavy drinkers showed higher blood-brain barrier permeability relative to moderate drinkers in the hippocampal cingulum bundle. So this is the white matter pathway that's the main input into the hippocampus. And there was no difference in permeability between moderate drinkers and abstainers or between heavy drinkers and abstainers. Again, our abstainers and heavy drinking groups were quite small. However, this result does kind of fit into what has commonly been reported with this J curve with alcohol consumption and particularly like cardiovascular health outcomes, where the heavy drinking group is really where we see that significant increase in risk. But then when you go from non-drinkers to moderate drinkers, there's either not much of an effect there, or sometimes people even see a beneficial effect with light to moderate drinking. Okay, so smoking, we didn't see any effects when we looked at the binary variable, whether someone has ever or never smoked. There was no relationship with blood-brain barrier. So this higher smoking intensity is related to higher blood-brain barrier permeability among men, especially in the white matter. One explanation for this involves known sex differences in cerebrovascular health risk. In particular, prior to menopause, the presence of estrogen in women is known to have vasoprotective and anti-inflammatory effects. So these premenopausal women may be, relatively speaking, protected against these outcomes relative to men. And this protection does appear to be lost after menopause. And while we don't have detailed timing information about the smoking history of our participants, everyone included in this sample indicated that they had quit smoking at least 2 years ago. So it's possible that many of our women smokers quit before menopause. Again, kind of speculative, but one explanation. Men also historically are reported to smoke more or use more tobacco products than women. In our sample, the average smoking intensity was higher in men than women, but this wasn't a statistically significant effect. So yeah, we're still kind of speculating here on exactly what this means. And then finally, with physical activity, this is the most surprising finding. So this time we saw an effect in women, but not men. So among women, regular strenuous exercise was associated with higher permeability in the parahippocampal cortex, the hippocampus, and the inferior longitudinal fasciculus. So why is something that we think of as positive exercise related to leakier blood-brain barriers in women? There's a few explanations here. The one that I'm currently the most fond of is that we may actually be picking up on increased vascularization in the brain. And there is evidence showing that exercise can induce neurogenesis in the hippocampus, and that's going to come along with increased vascularization. And if we're already looking at very low levels of permeability, perhaps the increased vascularization is going to have a stronger effect on our signal. Another possibility is that there is a point at which really vigorous and strenuous exercise might actually have detrimental effects. We can't tell that with our data. There also may be dynamic effects where transient increases in blood-brain barrier permeability could possibly reflect a beneficial thing. It's probably unlikely given that we're looking at gadolinium leakage, which is a large molecule, but nonetheless, there are multiple explanations here. I would not take these results to say that older women should not exercise, though, unfortunately. And then finally, the hours spent walking or sitting per day were not associated with permeability in the full sample, nor in sex stratified analyses. Okay, that was insights and advice. My last part here. So I wanted to share some insights I've gathered and advice that may be especially relevant for other trainees. The first is that your community, your mentors, your peers, your social networks matter a lot. And the second part might be slightly controversial, but I think that this matters perhaps equally or even more so than the details of the exact science that you're doing. And the reason that I believe this is because science is inevitably going to get hard. Experiments and analyses are going to fail or not go the way that you want them to. It's a very long, tedious process. Sometimes you'll apply for things and get rejected. It's going to get hard at some point. And when that happens, it's really important to be in a supportive environment with people who are going to help you work through that, support and respect you. And I think that we also do better science when we're in those supportive environments. So for those who are either now or in the future going to be in the process of choosing a lab or choosing a job, I always recommend considering the culture and the culture of the lab you're going to be working in and the people that you're working with, at least like on par with the exact science. And also, as scientifically curious people, I think we can get interested in a lot of things. So next, most, if not all of us, will experience imposter feelings, feeling like we're not smart enough, like we don't belong. And I think there are a couple strategies I've seen that have been really helpful in handling these feelings, both in myself or in my peers. The first is simply to say it out loud and talk about it with people. The more that you talk about this with your peers, you will inevitably realize that this is more of a universal experience and not really a personal, like isolated experience. I think this is partly just a nature of academia, but also just a side effect of advancing further along in whatever path you're on. The further along you get, the more you're surrounded by people who are really smart, really capable, really hardworking, more people who are experts. And we're going to compare ourselves to people, so it's really easy to feel like you don't quite fit in there. But if you're here and you're excited about your work and interested in it, you do belong here and you deserve a seat at the table. Then for those, especially those who do come from more underrepresented backgrounds, it can be helpful to recognize that these systems were not built for everyone and they were not built with everyone in mind. So from that perspective, some of you may be imposters, but that doesn't need to be a bad thing, right? It means the system wasn't built for you, but not that you don't belong here. And in fact, I think that means that your perspectives matter even more and your experience matters even more. So recognizing this could serve as some extra motivation to really advocate for yourself and fight for that seat. And then lastly, I think it's really important for us to openly express our excitement and our enthusiasm and our curiosity, both for establishing a nice working culture, but also for professional development, genuinely. The times that I've allowed myself to fully express my enthusiasm and curiosity for things, those have been the times where the coolest opportunities presented themselves to me, whether it was starting a new collaboration with somebody, being connected with a new mentor or peer, or getting the opportunity to teach a cool class. People remember when you are enthusiastic, and I think that they're going to be more likely to reach out to you with relevant opportunities.